Hello guys, welcome back to another episode of Learning with Isha. In the last video, I told about what is insulin, right? Why do we need insulin? Why this cattle and pig insulin was not enough for us, right? So now in this video, we are going to talk about two things. Number one is the structure of insulin. I told you it's a protein. We'll look more about that. Second part, I'm going to tell you how biotechnology is being put to use to make this insulin. Okay, let's begin with the first part first. So guys, insulin, first of all, I told you has 51 amino acids in its mature form. Please recall I used the word mature form, right? Since I emphasize this word, it means that there must be some other form, some inactive form, some immature form, right? That is called a prohormone. Pro and hormone. Hormone, why? Because it is insulin. Pro means because it is before my active form of insulin, before my hormone form of insulin. That is why it is called prohormone. This prohormone has 86 amino acids, whereas my mature insulin, the active insulin, has only 51 amino acids. This means there is a reduction in the number of amino acids, right? So there is some portion that must be present in the prohormone that would be getting deleted till the time my mature insulin is formed. That's exactly what happens, right? Let's see the structure of the prohormone. I have used three colors to represent three different chains. The purple one represents the A chain, the blue one the B chain, and the red one the C chain. Okay, the A chain has about 21 amino acids, the B chain has about 30 amino acids, and the rest is made up by the C chain. Guys, the C chain is an extra chain. It will be removed in this structure of the mature insulin. Then why is it needed? If I had to remove it in the first place, why am I spending energy on synthesizing this extra chain? Well guys, this has an important role to play. Amino acids come together to form proteins. But that is not the end of the story. Protein folding is a very important concept. If my protein doesn't fold together, if the tertiary structure of the protein is not formed properly, my protein is not going to be biologically active. So the C chain will help in the proper folding, in the proper coming together of the A chain and B chain to form the active insulin. Okay, it is removed, but before getting removed, it plays its own role. Okay, so my C chain, like I told you, is going to be removed. What I'll be left is with the A chain and the B chain. Now another important point to note is, for the insulin to be active, there are three disulfide bonds that need to be formed correctly. Disulfide bond क्या होता है? Sulfur and sulfur. A bond like this is called a disulfide bond. कौन बना रहा होगा ये bond? Give it a thought. Well, this insulin is a protein. It has amino acids. Now another question should come to your mind. कौन से amino acids में sulfur होता है? Which amino acids contain sulfur? Okay, there are two amino acids. Try to recall which ones. Well, they are cysteine and methionine. If you need to know more about these amino acids, please check out my video on amino acids. Well, cysteine and methionine amino acids are going to form this disulfide bonds. Three disulfide bonds will be formed. One will be within the A chain itself. Intra-chain disulfide bond. Intra means within the own chain. क्या positions होगी? 6th amino acid of chain A with the 11th amino acid of the chain A only. Okay. Now an interchain disulfide bond will also be formed between the 7th amino acid of chain A and the 7th amino acid of chain B. Another interchain disulfide bond will form where 20th amino acid of chain A with the 19th amino acid of chain B. So the bonds between the A chain and B chain will be called the interchain. Like we have an intrastate competition, right? Those of you who are keen in games would be knowing intrastate मतलब within the state, right? District level पे. लेकिन इंटरस्टेट का क्या मतलब होता है? लेट्स से पंजाब वर्सेस हरियाणा या कर्नाटका वर्सेस तमिलनाडु, things like this. Okay, so these are called interstate competitions. Similarly, interchain disulfide bonds. The one formed between the two amino acids in the A chain will be called an intra-chain disulfide bond. Okay. The process of the removal of the C chain and the formation of the mature amino acid having the A and B chain is called maturation. Yeah, pretty much like the name suggests. So this maturation is very key to the activation of insulin. Now I told you we'll talk about how biotechnology can be used to synthesize this insulin. Now we're trying to understand how we can synthesize this insulin in bulk. Okay, my aim to use biotechnology is number 1, to get contamination free insulin. Number 2, to not produce any allergy in my patients, right? So I'll take the sequence from the human, of the human insulin gene, okay, and synthesize it very large amounts. These are my 3 aims. What I'm going to do is, now here in my E. coli host, I see there are 2 types of genetic material present. This represents the genomic DNA of E. coli, okay. The genomic DNA is its own genome. What does this circular molecule over here represent? Well, it represents the plasmid DNA. What is the plasmid DNA? Well, plasmid DNA is a circular extra chromosomal piece of DNA. Okay, it's not part of the genome of the E. coli. It is extra chromosomal, extra. Okay, what is its advantage? Is that it is capable of making many copies of itself. How can it make many copies of itself? As it is self-replicating. What do you mean by self-replicating? Is that it can make multiple copies on itself. It can divide many times, 1 to hundreds, and then what will happen if my insulin gene is somehow added into it? Many copies of my insulin gene will also be produced. In the next step, what I'm going to do is, from the cells of a human, to avoid any allergy, what I'm going to take out is the insulin gene. In humans, the gene for insulin is called INS and is present on chromosome number 11. So I know the exact sequence. I'm going to take it out. What I'm going to do is I'm going to add restriction enzymes to my plasmid, cut out the insulin using the same restriction enzyme, and then ligate them together. What I will do is I will do a ligation using the enzyme ligase. Okay. Now what will form? My plasmid DNA, my insulin gene portion present over here. What will form is a hybrid molecule I will know as a recombinant DNA. Why is it known as a recombinant DNA? Well, because molecules of 2 different types are combining. Molecules from a prokaryote, the plasmid, is combining with the gene from a eukaryotic cell. So that is why it is called a recombinant DNA molecule. Now this molecule here is the vector, the plasmid, and it carries my insulin gene. So it is known as the passenger. Okay. Together, this molecule will now be added into my E. coli cells. इनका अपना जीनोम होगा, फिर मैंने उसमें ये ऐड कर दिया. यहाँ पे मेरी इंसुलिन जीन थी. What is going to happen is this plasmid will make many copies of itself, and as the E. coli cells divide every 20 minutes, lot of insulin protein is going to be produced. The DNA forms an mRNA, and the mRNA forms my protein. So my INS gene is going to form an mRNA, and eventually my protein insulin is going to be synthesized in very large quantities. So this E. coli cell is going to be grown industrially in what we know as vessels known as fermenters. These are very large vessels. चाय बनाने के लिए चाय का पतीला होता है, वैसे ही एक स्पेशल वेसल होती है जिसमें मेरे लार्ज वॉल्यूम में कंटामिनेशन फ्री कंडीशंस में ये बैक्टीरियल सेल ग्रो करेंगे. What I'm going to do is, after this insulin is produced, I'm going to take it out, recover it, and I'm going to purify it. Then I will have my purified insulin that will be bottled, and then it can be administered to the patient. So this was all about the synthesis of insulin using biotechnology. Thank you.